Welcome to another screencast where I'll be discussing an overview of gene expression. And before you embark on this, you should have a pretty strong understanding here of the phases of the cell cycle and a relatively strong understanding here of the structure and function of proteins and nucleic acids. So we're going to focus here on describing and explaining what we mean by gene, genotype, and phenotype. You have Bio 1 definitions for these, but we're going to advance those definitions and alter them just slightly. Be able to describe and explain, sort of in quotes here, the flow of genetic information. And then, most importantly, I think, be able to describe and explain relationships between terms that you already know here, DNA, RNA, polypeptide, and then transcription and translation. Again, these are from Bio 1. And then the relationship between other terms that come up and are thrown about here sometimes, triplet, codon, and amino acid. These things are related but are different. So let's get into this here. Here is, again, our cell cycle. For the previous part of the class, we were focusing mainly on what was going on in this part, in S and G2, and in the mitotic phase. Right? And so now we're going to be focusing what's going on primarily here in this G1 phase. And this G1 phase, this gap 1 phase, is primarily reserved for gene expression, right? Or you can think of it as that's what's going on here in this part of the cell cycle, where cells are doing their normal cell things, expressing genes, and we'll talk a little bit more about what we mean by that. But this is when, in the life cycle of a cell, these things are happening, or this is where we are in the life cycle of the cell. Protein structure review. Again, the proteins are made up of amino acids, and here is your basic amino acid structure, where your R group would be right here. So you need to be able to pretty much draw that forward, backward, inside out, understanding that this is what an amino acid is, and that you have different amino acids because of the different R groups, understanding that these amino acids are synthesized together via condensation reactions here, and creating then what is known as a polypeptide, right, or a string of amino acids. And this amino acid sequence is extremely important. What makes an amino acid different from one another, of course, again, are the 20 different side chains shown here. Again, not supposed to be memorized, but there's 20 different amino acids can be placed in many, many different orders to get then the different primary structures, which then yield the different tertiary and or quaternary level structure proteins due to folding and interactions between the R groups. Okay, so the amino acid sequence is extremely important here, because this amino acid sequence will determine how that thing folds up, and that thing folding up in 3 dimensions does have some sort of functionality to it. For example, we have different types of proteins here. Some of them are enzymes, like this alcohol dehydrogenase, right, is an enzyme. We have proteins like collagen that play structural roles in cells. You have very simple amino acid-based proteins like epinephrine here that are hormone and neurotransmitter-based or have function to them. And then here's a protein that most of you are probably familiar with, is insulin, helping to get blood glucose into or out of cells. So lots of different functions, lots of different shapes and sizes to these proteins. And again, because of what is varied here, and that is, again, the amino acid sequence. So these amino acid sequences, these proteins, right, are ultimately what determine a cell's phenotype. So the phenotype, you're used from Bio 1, used to thinking of it as like the color of the eyes or something like that. When really now at this level, we're talking about phenotypes being some sort of a protein, whether that protein is structurally correct or not, structurally functional or not, will often yield whether that cell performs properly, improperly, differently than say another cell. So phenotype now is at the protein level. Also reviewing here DNA structure and the RNA structure from previous classes. Again, this DNA is anti-parallel, right? Double-stranded molecule over here. RNA over here, single-stranded polymer. of nucleotides, and it's the sequence of these nucleotides that we're going to be sequence of nitrogen bases, sequence of the nucleotides that we are going to be most concerned about. Okay? So there's a relationship between the sequence of DNA nucleotides, the sequence of RNA nucleotides, and the sequence of amino acids in the protein. So here is a graphic that shows that. So we're defining now genes as a segment of DNA, sometimes RNA, depending on the organism, but a segment of DNA sequences that now how we say this is that we say that it codes for a polypeptide. So a gene is a segment of DNA that codes for a polypeptide. And this is what this looks like. So if we take this gene, if we take this little sequence of this one gene and look at it here, we see that we have a sequence of nucleotides, adenine, cytosine, cytosine, adenine, adenine, et cetera, so on down the line. This is known as the DNA template strand. Notice that we're only looking at one strand of this. We're going to look at it from a 3' to 5' direction. And we can break this DNA template strand down into groups of 3 that we call triplets. So a triplet is synonymous with DNA, so 3 nucleotides in a DNA strand. When the cell is going to now convert this, well, it uses this DNA as a template. It doesn't convert, it doesn't turn the DNA into RNA, but it uses the DNA as a template in a process called transcription to then make groups of 3 mRNA nucleotides, what we refer to as a codon. So triplets are complementary to codons. Notice that this RNA sequence right here is also running 5' to 3'. And we can break this down into groups of 3. And again, this should be a review from you from Bio 1. These codons then, again, are synonymous with mRNA. Right? So you wouldn't say that, you would not call this a DNA codon. You would call this a triplet, and then you would call this a codon down here. So these codons then are the code for the ribosome then to translate this mRNA. Again, the mRNA is not turned into this amino acid sequence down here, but rather used as a template for the ribosome to then start assembling amino acids. And this would be an amino acid tryptophan, and then this codon here, UUU, codes for phenylalanine, and this codon codes for glycine, and so on and so forth. Right? So this is the basic flow of genetic information from DNA to RNA to polypeptide. Okay. In older textbooks and older circles, you may also see this or hear this referred to as the central dogma. Right. That term not used as much anymore. But again, the flow of genetic information from a DNA sequence transcribed to an mRNA sequence, and then that mRNA sequence then translated into an amino acid sequence. So if we go back here, if we look again at this gene concept, this segment of the DNA sequences that codes for a polypeptide, this sequence right here can also be referred to as the genotype. Which types of genes do you have? Meaning, which sequences do you have? Right? So if you're talking about someone's genotype, right, you're referring to the specific set of sequences of DNA. Right? So genotypes then code for phenotypes. That is the amino acid sequence that is going to then determine how that protein, that polypeptide, forms up. So there is the basic general overview of gene expression. So I said that this was the central dogma, and we said that this is how information flows. This is also referred to, and you hear me say this very often, is what we mean by gene expression. So this whole thing here is gene expression. Expressing the genes means to transcribe them and then ultimately translate those RNA sequences. So here we see gene expression occurring in prokaryotic cells as well as eukaryotic cells. In a prokaryotic cell on the left here, here's our DNA sequence, and it is going to be transcribed into an RNA sequence. Then that RNA sequence will be translated by ribosomes out here in the cytoplasm. And the ribosome will use that RNA sequence to then assemble nucleotides. Sorry, strike that. We'll use the RNA sequence to assemble amino acids together to create the polypeptide. So this is gene expression in prokaryotes. In eukaryotes, we have this very similar pattern here where here's our DNA. It's going to be transcribed into an RNA molecule, but there's a little bit of extra stuff that's happening here in the eukaryotic cell. We get a little bit of RNA processing, which we'll discuss further in another screencast and in class. Suffice it to say, though, that you end up with this RNA molecule that will then be translated by, again, ribosomes to generate a polypeptide here. And this polypeptide, of course, is amino acid sequences. One thing I will note, obviously, is that eukaryotic cells have this nucleus and this nuclear membrane, this nuclear envelope that physically separates the transcription processes and the RNA processing from the translation that happens out in the cytoplasm. If you can compare that with a prokaryotic cell, as soon as you get transcription happening, you get translation happening. There is no nuclear membrane or nuclear envelope there to block that. It's sort of one right after the other. One final note here is that you've also heard of other types of RNA molecules. We've been dealing primarily with this mRNA. In other screencasts and in class, we will talk about the roles of tRNA and rRNA. You have invariably heard of those in Bio 1 and may understand a little bit about what they are, but we're going to broaden those definitions a little bit, and then also throw at you that there are other types of RNA out there, right, that are new here probably to you. So small nuclear RNAs, SRP RNAs, and we'll get to those in a little bit more. But I want to introduce this to you here now and let you know that there are more than just one type of, or more than the three types of RNA that you're classically learned in Bio 1. Finally, you've seen this, the Codon Table. And this Codon Table is a human construct that allows us to express DNA sequences, transcribe, translate DNA sequences into RNA sequences. And really though, this Codon Table is for the codons, right, to know which codons code for which amino acids. Right? So you look at the first base here, you look at the second base there, and the third base over here. So, for example, if I gave you a DNA sequence that was CCT GCA, so these are the triplets. What are the RNA codons that correspond to this? Well, I would make this C would be a G, this C would be a G, this T would be an A, this G would be a C, this C would be a G, and this A wouldn't be T but would be U. So these are my two codons. So GGA, what does GGA code for? Well, here's the first base, G. Here's the second base across the top, G. So I know that all the ones that are G, G something are down here. And I look at the third base, right? So I need A, GGA right here. So GGA, there's the codon, tells me that it codes for glycine. Right? And so that's glycine. CGU then, there's C, here's G, and here's U. This one codes for arginine. This would be ARG. So some examples there and an overview. So go back. Can you describe and explain here now? Gene, genotype, phenotype. Describe and explain the general flow of genetic information from DNA to polypeptide. And then clearly describe relationships between DNA, RNA, polypeptide, and then what these processes here, transcription and translation, get you, and then the relationship between triplets, codons, and amino acids. Thanks for listening. Bring your questions to class. I'll see you there.